All right, yes, we are all set up. So today we're going to talk about the peripheral chemoreceptors. Um, this is a topic, it's the carotid bodies, of course. This is a topic that has been neglected through the years somewhat, but has come into prominence lately. And that's largely because it's being blamed for an excessive sympathetic activity, which causes high blood pressure. And some people are advocating that we simply should remove the carotid bodies and people would get better. This is not so good because you lose any sensitivity to hypoxia that you might have. The other aspect of the peripheral chemoreceptors that to my mind, has been neglected, and I've been trying to do something about it for years, but so far, no. The textbook idea of the peripheral chemoreceptors is that they are hypoxia sensors, and they are only come into play when we get hypoxic. And that's how they're played. Oh, by the way, they're also sensitive to PCO2. And indeed, if you get hypoxic, your sensitivity to PCO2 increases. That's the textbook view. And it's a sort of hypoxia sensor-centric view, which I think is incorrect, as we'll see as we go on through here. I think it's, in fact, a receptor very much like the smooth muscle of the cerebrovasculature, RTN neurons, although in this case they don't fire, they simply release transmitters, but they are sensitive to their intracellular hydrogen ion concentration. And what hypoxia does is it interferes with the regulation of that level of hydrogen ion concentration, as we'll see as we go through. So what about some history first? And I've taken more or less the abstract from this 2009 article, which deals with the history of the carotid body, of its discovery, and so forth, the major players in its discovery in red type here in this slide. I'm not going to go through and read it, but it turns out that one of the students of de Cajal, Fernando de Castro, was the person that really used the histology to investigate the structure and the innervation of the carotid body. Now, as for the physiology of what it does, that will be Heymans et al. And his son. And indeed, they were awarded the Nobel Prize in Physiology in 1938. And so they're the people that find out that, this is what it does. And we'll see what it does as we proceed. So here we've been reading the annual review here. I hope you gained some benefit from having some of the areas highlighted that I felt were important, and leave some of the detail alone in that article. And we see the picture of where we are. Can somebody let these people that are waiting in the lobby in? Can I do that? I don't see anybody waiting in the lobby. They have let them in. All right. All right, we will continue. The carotid body itself, where is it? It's in located at the bifurcation of the carotid artery into its internal and external branches, right there in the neck. There is a sinus nerve that goes into it, that is a sensory nerve that joins in to make the glossopharyngeal nerve. That ends up relaying in the nucleus tractus solitarius in the medulla. And if we look at some of the cells that are inside, we have this sort of glomal cell and its neighbors into a sort of unit, if you like, that is going to be our sensor. And we have many blood vessels going through it. We have the glomus cells itself. We have some type 2 cells, which are sort of like glial cells, are in the brain, but this time they're in the carotid body. And it is the glomus cell that is our sensor. And what does it sense? Well, in this case, it's supposed to sense hypoxia. And that may also be sensing hydrogen ion concentration 2 and PCO2. It senses these in the arterial blood in the vessels passing through. And what it does is it releases transmitters which excite the sensory nerve. And so there's the chemosensory synapse here that is causing the sensory nerve to send discharges up towards the rest of the brain. And that is basically how it works. Lots of experiments have been done to find out what is the transmitter here that is used. Looks like ATP is coming out as the major player, but there are many other interesting molecules that are also involved in this. And that kind of study has gone on over a large number of years, where the sort of concentration of physiologists has been on trying to find out how oxygen is sensed, rather than taking its functional aspect and recognizing how important it is in various autonomic regulations throughout the body. So, transmitter release. We've got various things that will cause transmitter release. Yes, it is sensitive to the arterial hydrogen ion concentration and the PO2 and the PCO2, presence of lactate. It will sense hypoglycemia. There's something new and different. And we see, as we have done before, in the smooth muscle tone and the RTN central chemoreceptor model that I proposed, that it is calcium ions that are the mediator, that mediate the release of transmitter here. Same as they mediate the vascular smooth muscle tone and the excitation of the RTN central chemoreceptors, I think. He does show you, is ATP signaling involved? Well, my good friend Julian Payton here, he's a major player in this, and you can look up lots of his papers. If you're interested in the aspect of control of high blood pressure and possible removal of the carotid bodies, that would be the name to look for. And Colin Nurse, our Canadian investigator here, has also published reviews on the paracrine mechanisms in the carotid body. Well, here's a carotid body slice. I never did quite identify where it was from. But here we are. Yes, this is what is thought to be the thing, hypoxia. But also works for hypercapnia, sees lactate, sees hypoglycemia. And also note that if you make this very acid, the response to hypoxia increases. This is tremendously acid. This is way beyond the normal range of pH in tissues. So that's the slice apparently can tolerate this. But there we are. There is transmitter release. These are release recordings of the responses of the transmitter release. And so there are all of the things that it responds to. So what other aspects are there? We should point out that it has the highest blood flow per tissue volume. And therefore, there is no gradient, so to speak, between what the glomus cells are seeing and what's actually in the blood, apart from membranes in between. It therefore tastes the blood the PCO2 and the PO2 and hypoglycemia, et cetera, leading to the brain. In other words, this is the early warning sensor because it's going to be protecting the brain. And remember, the whole system is designed to protect the brain and maintain the brain milieu. The blood flow in the carotid body can be controlled possibly by autonomic efferents. They may adjust the blood flow through it and therefore adjust the sensitivity. This involves nitrous oxide and vascular control. Here's a reference for it. The other aspect of the carotid body is that it is plastic. You get hypertrophy during prolonged hypoxic exposure. And there's a nice article by Prem Kumar and Nirandi Prabhakar here that is very good for a summary of that. And again, here's Julian Payton again. The carotid body stimulates the sympathetic system. Now, Daniel Kerr and I started to study this too. And we looked at the carotid body and the sympathetic system here, published here, and we used rebreathing methods, which we'll talk about next week, in order to keep the partial pressure of oxygen constant at either a high level or at a low level here, 150 millimeters of mercury, 50 here. And we use rebreathing to introduce a ramp stimulus of CO2 that slowly increases over time over the second. So this is about 5 minutes worth here, 300 seconds. And so CO2 is slowly rising, and yet we maintain the partial pressure of oxygen constant. What happens? Well, one of the things that we can see is that the blood pressure may increase. In some people, it goes along constant and then suddenly increases. In this person, it seems as though it's slowly increasing all the way through. And the other thing that happens is that the tidal volume. At first, nothing much happens, but we do reach a threshold point where we increase. But here's the important part. We're talking about the sympathetic system. We see that the sympathetic system, these burstings of activity in muscle sympathetic nerve activity, these bursts increase in their frequency and in their amplitude. It is even exaggerated more when we use hypoxia and the same ramp increase in CO2. We see a similar rise in blood pressure, but ventilation increases much more, has an earlier onset of the threshold, and the same goes for the sympathetic. So the carotid body, as I said, is now thought to be involved in high blood pressure because the carotid body is overactive. Therefore, maybe we should take it out. Here's, again, from Julian Payton's lab. This is very interesting experiments to show you, if you like, a sort of an electronic connection diagram. What they have proposed is that there are carotid body gloma cells that activate nerve endings that are specific in their targets as to where they go in the nucleus tractus solitarius and where those get relayed to. In other words, they make it like an axonal ribbon cable of connectivity. This is a very interesting idea that it's not just that the nerve ending firing pattern, increase in frequency of firing, increases for everything, but it goes to a particular point where it can do the most good. Perhaps into rhythm generation here, also perhaps into the retrotrapezoid nucleus, as we saw in the central chemoreceptors. It goes down to control some of the muscles in the airway and various other parts of the body. It is a major player in autonomic control. And that is becoming more and more apparent as we study it. And we can see here, with the same paper, this diagram as well. Stimulate here with this lightning bolt, and what kinds of things can we affect? Phrenic nerve activity and ventilation, sympathetic activity, we get bradycardia off that, and tachycardia, heart. So we have cardiac, vascular, respiratory, metabolic, renal, gastrointestinal, hematologic, increasing hematocrit. All of these are involved. Do you want to take this body out and deprive your patient of this? I don't think so. So, some more ideas. It is the only hypoxia sensor for respiratory control. If you remove it, there is no response to hypoxia. Honda, who came to dinner at my house, no, came to lunch at my house at one stage, studied this in carotid body resected humans. I think this is the very early studies, and there are current studies. Here's my friend Julian Payton again, and in a more current view, again, what are the responses to hypoxia and hypercapnia following bilateral carotid body resection in humans? So that's a good paper to look at if you're interested in that theory. There have been many proposals. As I said before, people have concentrated on finding out how does it sense oxygen. And these folks have done a review at a rattlebag of molecular mechanisms. In other words, there are quite a few ideas out there. Perhaps the involvement of carbon monoxide and hydrogen sulfide as transmitters. Yes, Nurendi Prabhakar has published a review on that. What about an oxygen-sensitive potassium current that therefore controls the membrane potential and internal calcium ion concentration from Gonzalez and company, another name that one should look for when you're considering investigating the carotid body. And from our current review that we were assigned the mitochondria to membrane signaling model of acute oxygen sensing, which we will look at in the next slide. But as I referred to before, I think they're all wrongly directed. They're looking for a hypoxia sensor. I don't think there is one. I think that the carotid chemoreceptors are primarily hydrogen ion concentration receptors, in that they sense changes occur because of their intracellular hydrogen ion concentration changes. And I think that hypoxia simply impedes, there's a missing I there, their normal function by acting as a fail-safe control, exactly like hypoxic inducible factor control system. That's a fail-safe system. You've read the paper. You obviously know what I'm talking about. So here's the carotid body sensing. I thought we had to put it in there to show the kinds of things that are going on. We think there are calcium channels. Oxygen is being sensed. These are all the kinds of complicated things that are going on across the plasma membrane. Here's the potassium channels, which may indeed be sensitive to the hydrogen ion concentration environment and so forth. So if you are at all interested, you can pursue this kind of aspect. Now, what about the discharge of those afferents? I couldn't find much in the way of information about those, except in Bob Torrance's review, which was published in a book, and also in this Advances in Experimental and Medical Biology. I used to assign this as the reading, but it is very dense. The ideas are fascinating, He has a way of explaining them that is rather difficult for the modern student to comprehend. And so I have left it out now and simply tried to put his ideas into this talk here. But what he has here is the effect of alveolar PCO2 upon the discharge in a single carotid chemoreceptor fiber at different levels of O2. These are the different levels of O2. You can see we're getting hypoxic. This is the frequency of firing of that nerve fiber. And so you can see there appears to be more or less a linear response to the increase in PCO2, but the sensitivity, the slope of this line, is affected by the partial pressure of oxygen, so that it gets more and more sensitive to PCO2 as you get hypoxic. In other words, this is the early warning for asphyxia, because in asphyxia, PCO2 rises and PO2 falls, and they both occur. And when they occur together, we get the maximum frequency of firing out of the carotid body saying, do something, breathe. We can look at this information another way, if you like, if you plot PO2 and the firing frequency here. And again, this is the steady discharge in impulses per second of two single carotid chemoreceptor fibers at a constant PCO2. Typo in the figure here, by the way. This comes from a thesis, an Oxford University thesis, because that's where Torrance was located, in 1974 from Hayes. And each of these fibers has the same kind of hyperbolic, rectangular hyperbolic response that as the PO2 decreases, we find that the frequency of firing increases. Now, other aspect of that discharge, those were the steady state responses. What happens, and again, this is from Torrance's Prolegomena, what happens if you add a square wave of PCO2 and look at the discharge frequency here, that's an F. It suddenly increases, but then it decreases. And then when you take it away, take the CO2 away, it drops down below and then comes back. And now if you look at this another way and say, all right, we'll do a steady PCO2 increase and hold it there. What do we observe? We observe the frequency goes up and then it declines. Depending upon the PO2, that decline in the frequency of firing over a fairly short period of time here is more and more. So that if the partial pressure of oxygen is quite high, by the time you've reached a steady state, it's almost not responding to PCO2 at all. And we'll see how important that is next week, actually, when we study the whole system together. So here are some of the gems of knowledge out of Torrance's Prolegomena. He talks about the sudden change in CO2, telling you something about the mechanisms of the response and the two components. First, a step up that is fast, and the height varies with the size of the stimulus, PCO2. But that height is independent of whatever PO2 is. And then there's an exponential decline, or we might call it an adaptation of the response, and that degree of adaptation is dependent upon the PO2. So as we saw on the previous slide, that means if the PO2 is high, then yes, you will get a sudden increase with your PCO2 step up, but it will decline down to a steady state in high PCO2, which is virtually no response at all. And here we have another little paragraph from that. And so I underlined it. All that oxygen ever does, it reduces the size of the response to CO2 that CO2 would otherwise be producing. So that, I think, leads into what I'm going to tell you how I think it works. And that I got these ideas from this. It's not a dreary little CO2 receptor brought to life by hypoxia. It's very excitable CO2 receptor, and it's calmed down by oxygen. Now, the other thing that I haven't reviewed here, other than to mention it here, is that Torrance had a bicarbonate hypothesis to explain a carotid body sensing. And if you base it on hydrogen ion concentration, it leads to, if you like, an acid-base hypothesis based on the Stewart approach. And this is my hypothesis. I think that the carotid body intracellular hydrogen ion concentration is maintained at a constant level. It's regulated. All cells try and regulate their intracellular hydrogen ion concentration. But if you add CO2 into the arterial blood, which is being seen by the glomus cells, then you are going to disturb the intracellular hydrogen ion concentration because CO2 diffuses everywhere. So the regulation of hydrogen ion concentration, if you have a regulator for it, that requires energy. And therefore, if you take the energy supply in terms of oxygen away, then hypoxia will reduce the efficiency of that maintenance of the hydrogen ion concentration. Therefore, it will lose out. Hydrogen ion concentration will increase because of the CO2, and therefore calcium, and therefore discharge. So that led me to propose that the oxygen supply is fail-safe. There's no specific hypoxia sensor. It's just the withdrawal of the energy supply to the CO2 or hydrogen ion concentration sensor. And this, of course, comes from the HIF system. So here is my fail-safe model. You've been reading about it, but we may as well go over it here. That hydrogen ion concentration, as we've seen from the Stewart approach, is a function of the PCO2 and the strong ion difference inside the glomus cell. We have PCO2 will affect that. PCO2 from outside in the arterial blood, and we have PO2. We have ion pumps that control the strong ion difference inside, and they require oxygen. So this is the simple mathematical model. It's virtually linear, except for this inverse here. The transmitter release will vary directly with the hydrogen ion concentration because the calcium ion concentration will vary directly with it, hypothesized. And the frequency of firing, if we wanted to convert it, would be a simple linear response here. That carotid sinus nerve stimulates pulmonary ventilation to rectify the problem and try and lower PCO2. So we go through the steps here. Hydrogen ion is regulated to maintain cellular function by altering the strong ion difference. External CO2 challenges it. Ion exchanges across the membrane try and restore it, but they require energy and oxygen. If the oxygen supply fails, PO2 is decreased. Consequently, hydrogen ion is not well regulated and increases with the PCO2, and that increases calcium, causing the release of transmitters, etc. So that outlines the entire model. It's quite a simple model. That may or may not be true. Now, if it wasn't true, I wouldn't be able to produce these diagrams that would match those observed. So here is the PO2 changing on getting more and more hypoxic, and the frequency of firing in response to CO2 linear, but the slope sensitivity increases with hypoxia, exactly as this is the reproduction of the data in the prolegomena from Hayes' thesis. And so, yes, that looks the same. So it is not impossible. The same goes for the idea that we have a hyperbolic relationship between PO2 and the firing frequency here, and that this varies with PCO2. And this is the kind of responses you see as CO2 increases more and more, we get a greater and greater impulse response to the lowering of PO2. I also took a look in the model to see if it would do the transient responses. And yes, it does. So if you make the PO2 150, yes, you get a big jump of the when CO2 is applied, and then that response falls down. If the PO2 is 50, it doesn't fall down so much. So there's the steady state responses to CO2 becoming apparent when you begin to be hypoxic and getting more and more vigorous. So my final words on this are that this is a model. It does produce observations that show that it works in a realistic way to control the impulses coming out of the carotid body. But I do emphasize that it means that the model is only a possible truth. So somebody out there hopefully will take it up and see if they can prove or disprove that the theory is correct. It does show you that, and what is apparent from the various experimental findings, that the carotid body is sensitive to the respiratory aspects, arterial PO2, PCO2, there's arterial values, and arterial hydrogen concentration. And do note that it is not sensitive to content. So that if the content of oxygen carriage decreases, such as in carbon monoxide poisoning, our early warning system does not work. It's only when you become hypoxia and the partial pressure of oxygen goes down that it works for us. So it's an asphyxia sensor. The gloma cells do release transmitters that activate the carotid sinus nerves. Which ones are the most important? Are they all there? Do they all do something different? There is a response to the PCO2 change or the hydrogen concentration change that has a threshold. And below that, there is no response. And note that this response to PCO2 and hydrogen concentration is modulated by PO2 such that hypoxia reduces the sensitivity to PCO2 to insignificance. So these are known. How it goes together is not known, but proposed here in this model.